Okay, first of all, I'd just like to thank you for giving me the opportunity of presenting my work here today. What I'm going to talk about is visualizing protein aggregates at the single molecule level. So just to start, I'd just like to thank my research group and the people involved in this research and also the funders. So we're a diverse team of biologists, chemists and physicists across all different career stages from lots of different places around the world. So neurodegenerative diseases, why am I interested in these? What's the size of the problem? Well, 1 in 3 people alive today will develop a neurodegenerative disease. And the annual cost of these is larger than that of cancer and heart disease combined. So it's around £500 billion worldwide per year. So that's about 1% of global GDP. The majority of that cost isn't spent on research, it's spent on actual care for people living with neurodegenerative disorders. It's the only leading cause of death, which is increasing year on year. As we have an aging population, this problem is only going to get worse as more and more people develop these neurodegenerative disorders. Yet despite this, we have no cure, no prevention, and only limited disease modification. So what's the reason for this? Well, neurodegenerative diseases are one of a new type of diseases, and they're related to this process called of protein misfolding and protein aggregation. So if you see here, we have our functional protein. So these are meant to represent different conformations of a protein that can act and function. So it's monomeric. But what can occasionally happen is we can have misfolding of this functional protein and then clumping together to form these so-called protein aggregates. So you can see here, we have these smaller clumps of protein. These can then go on and develop their structures further until we eventually get these amyloid fibrils here. And these are what you would find in the amyloid beta plaques or in the Lewy bodies found in Parkinson's. One of the major problems that we have is that by the time that visible symptoms are present, most of the damage has already occurred. In fact, it's occurring decades prior to the formation of these symptoms. So ideally, what we would be able to do is we'd like to be able to detect and probe the biophysical status of these developing protein aggregates in vivo prior to symptomatic onset. If we could do that, we could then diagnose earlier, and we could also test therapeutic interventions at an earlier stage. So how are we going to do this? Well, at this point, I like to quote Richard Feynman here, who won the Nobel Prize in Physics. And what he said at a meeting in 1959 is that it's very easy to answer fundamental biological questions. You just look at the thing. Unfortunately, the present microscope sees at a scale which is just a bit too crude. Make the microscope 100 times more powerful, and many problems of biology would be made very much easier. I exaggerate, of course, but the biologists would surely be very thankful to you, and they'll prefer that to the criticism that they should use more mathematics. So this quote kind of sums up my work. So my group design and build and develop microscopy techniques that allow us to look at individual molecules at the super resolution level, so at the nanometer length scale. And by using those to look at protein aggregates, we're therefore able to shed light onto the process and hopefully develop diagnostic processes and tools for these disorders. So what I'm going to talk about in this presentation is some challenges that we've overcome for oligomer detection. So from that quote, the first thing we want to do is we want to be able to detect individual molecules. If we can detect those individual molecules, what we can do is we can distinguish the oligomers from those functional monomeric protein. We want to also make it label-free. If we don't need to have a covalent tag on our protein, then we can detect oligomers present in biofluids or biological samples and not rely on just purified protein. We want specificity. We want to be able to definitely say that the aggregates that we detect are formed from a certain protein. And finally, we want to image those aggregates at a relevant spatial resolution. So going on to the first challenge, the detection of individual molecules. So we can achieve this using the instrumentation that we build in our lab. So most of the instruments that we use are these so-called total internal reflection fluorescence microscopes, seen here. So this is just in one of our dark labs, and we have a series of lasers that are aligned into the back of a conventional microscope. And what these allow us to do is to detect individual molecules, as seen here. So by using TIRF, or total internal reflection fluorescence microscopy, TIRF microscopy, we can detect single molecules. And at these molecules here, each one of these white spots corresponds to an individual molecule. And in this case, it's the protein alpha-synuclein. And alpha-synuclein has been modified with this organic fluorophore. And every molecule of alpha-synuclein is tagged with one molecule of the organic fluorophore. So there's a one-to-one labeling. And what you can see here is that we have lots of different spots, each one an individual molecule. If we look at these more carefully, you can see we have a bright spot here and a dim spot. So already just by looking at individual molecules, we can say something about the state of the protein. These dim spots here correspond to the functional monomeric protein, i.e. there's only one di molecule present, so it's quite dim. However, we have brighter spots, and these are the protein aggregates, so those clumps of proteins, the oligomers. These are brighter because there are more molecules of synuclein present there, so we have more molecules of the fluorophore, and therefore a higher fluorescence intensity. So tagging molecules and imaging them on fluorescence microscopy, or using fluorescence microscopy and turf microscopy, we can already distinguish oligomers from fibrils. So we can detect individual molecules, and that allows us to see the subpopulation of oligomers here. But if we really want to be able to look into biological samples, we can't rely on conjugating our protein to a tag or an organic fluorophore. In the previous case, we had to purify a modified version of synuclein where it had a cysteine incorporated so that we could label the cysteine 1-to-1 with an organic fluorophore. But we obviously can't do that in alpha-synuclein from patient samples, such as cerebrospinal fluid. So the way we got around this was using this so-called single aggregate visualization by enhancement technique. And in this approach, what we do is we make use of this dye, thioflavin T. So thioflavin T, or THT, has been used for many decades to visualize protein aggregates. But rather than looking at them using microscopy, it tends to be used in a fluorescent plate reader. So when THT is free in solution, there's rotation about this single bond, and that leads to quenching. So we don't see any fluorescence from THT. However, what it can do is it can sit in between the beta sheet, the ladders on this beta sheet here, this extended beta sheet structure. And when it does so, this rotation is prevented. We don't have rotation then, and we get a fluorescence switch on. So in a plate reader experiment, what we would see is we would have our monomeric protein would have zero fluorescence, but as the protein aggregates, we would then get an increase in the fluorescence intensity. And that would then allow us to detect the amyloid load that's present in the sample. Now, what we decided to do is to actually use this thioflavin T, but rather than detecting its bulk fluorescence on a plate reader, we decided we'd use the turf microscopy technique once again. So we take our sample of aggregates, and this time it doesn't have to be labeled, and we just introduce thioflavin T, and then we image whatever thioflavin T is bound to. And when we do this, we can then detect protein aggregates. Here is just an example of some CSF samples that we've measured, and you can see here we have a healthy control, and we have a few of these bright spots here which correspond to individual oligomers of any protein. We can't say for sure it's alpha-synuclein. When we take the Parkinson's disease sample, we see many more of these bright THT active spots. If we then compare a series of different Parkinson's disease samples with healthy controls, we find there's a statistically significantly higher number of protein aggregates in the Parkinson's disease sample compared to the healthy control. So the advantages of this technique now are that we can visualize these proteins at the single molecule level. We can specifically detect oligomers and fibrils and not see the monomeric protein. And we can also then visualize these samples, visualize these oligomers in biological samples and not have to label the sample of interest or the protein of interest. However, there's a huge disadvantage to this technique. And first of all, it's not specific to the different disease. So these aggregates are likely to be alpha-synuclein containing because we're looking at Parkinson's disease, but they may also be formed of tau or amyloid beta or other aggregating proteins. So it's not specific to the protein of interest. The other problem that we might have is that THT might not detect the earliest oligomeric species. So as I mentioned previously, it has to bind to this amyloid structure where we have these extended beta sheet structures here. But the very earliest oligomers, which might be the damaging ones, might not necessarily bind THT, and so we don't necessarily detect them using this approach. So we've detected individual molecules. We've then developed our approach to allow it to be label-free, so we can look in biofluids. The next thing that we want to do is to increase the specificity of detection, i.e. we want to be sure that the protein we're looking at, or the oligomer we're looking at, contains the specific protein that we're interested in. So at this point, I'm just going to introduce a technique that was developed by T.J. Haar, and it's called this simple or single molecule pull down. So in the previous approaches that I've described, I didn't really have to do much to the cover slip, which is visualized here, in order to get the aggregates to be present there. all I did was I added any protein such as polyl lysine, which makes a positive charge on the surface, and then the aggregates would sit down. But that isn't very specific. Now, what this simple technique does is we can actually passivate the surface. We can prevent molecules from binding to it. And we do this by having a polyethylene glycol surface or a PEG surface. So we can passivate the cover slip with PEG, prevent non-specific binding of any molecule. We can then have a small percentage of those PEG molecules biotinylated, and we can then use streptavidin as a sandwich to pull down whatever we want to them with a biotinylated form. So we have a passivated surface which won't bind anything, and then we have a streptavidin layer on top. We can then introduce a biotinylated capture antibody. So now we can pull down a specific protein, and then when we introduce our aggregates, we would have our protein of interest bound to that. So in this case, it could be alpha-synuclein. So the antibody would recognize a specific epitope on the alpha-synuclein, and then we would pull down specifically alpha-synuclein. So we'd have monomeric protein here and a oligomeric protein. To detect the alpha-synuclein, remember it's unlabeled, we would then use a second antibody. So we have labeled detection antibodies. So these once again can be labeled with organic fluorophores, like our alpha-synuclein case, and we would then look at the intensity of these to determine what's bound. Now, we could determine from the intensity whether there's a monomeric protein or an oligomeric protein just by looking at the intensity. However, that's not that accurate. We might have some very small oligomers which have only a very few epitopes present, and they might look very similar in intensity to the monomer. So we might not necessarily be able to tell the difference between our monomer and oligomer. So what we decided to do is to improve this technique further by introducing a second color. And we called it the so-called staple technique. So in staple, which is single molecule two color aggregate pull down, we now know when we have a monomer versus an oligomer just by looking at the different colors. If we have monomeric protein, it can either bind the green antibody or the pink antibody, which both target the same epitope. However, if we have an oligomer, we have many copies of that same epitope, in which case we can have both colors bound. So we can have a green antibody and a pink antibody bound at the same time. So just by looking at the coincident signals in our two-color approach, we can then distinguish oligomer from monomer. So we have a way of being able to now specifically image oligomers of a specific protein. And this was published last year in Science Advances. And this work, I should say, was funded by UCB Biopharma. So what does this look like? Well, we used antibodies provided by UCB to target alpha-synuclein, and this is work done by Craig Layton, Rebecca Salib, and Jian Li. And if we have monomeric protein, we have our alpha-synuclein antibody here and our two detection colors. With monomeric protein, we can see we have some pink spots and some green spots. These correspond to the monomeric protein. There's no coincident spots. However, when we have aggregated alpha-synuclein, you can now see that the pink spots and the green spots overlap. And that's because we have multiple copies of the antibodies bound. So all we then need to do is to count the number of coincident spots, and that would allow us to determine the number of protein aggregates that we have present. So the staple idea works. We can get single molecule two-color aggregate pull down. If we look at how well the cover slip works, so in this case here, we have a non-treated cover slip with the antibodies present. You can see there's lots of spots everywhere with just the antibody alone. With the simple surface, we can see a few spots that correspond to just antibodies sticking to the surface, so there's not complete passivation. However, for the staple approach, we can actually use the advantage of having two colors to remove the background noise as well. Because we're only counting coincident events, the chance of having two antibodies bound in the same place is very low. So we get way fewer events detected when we use our staple approach. So this is just shown here. Non-treated, you can see there's a significantly larger proportion of molecules on there, so we count many more molecules. With the simple, we see fewer of them. However, with the staple, we have very few coincident events. So we passivate the surface and we can really specifically detect the molecules. Here we then wanted to compare how good we are at actually detecting oligomers. So these are proteins that are provided by StressMark Biosciences. And you can see here when we have monomeric protein, if we use our save via thioflavin T, we do see some aggregates that are present, but not many. If we look at our oligomer, this is a purified kinetically trapped oligomer from StressMark. you can't see many events at all. So this oligomer doesn't seem to be thioflavin T active. However, when we take our fibril sample, also provided by Stressmark, you can see there's many thioflavin T events that are active. So we can detect fibrils, but we can't detect oligomer, and we see a few events for the monomer. If we now take our staple approach and look at the same samples, with the staple approach, we don't see many monomer. There might be some aggregates there, so we do see a few aggregates present in the monomeric protein. But now with the oligomer, we see many, many events. So even though these oligomers aren't thioflavin T active because they don't have extended beta sheet content, they do contain many oligomers, and we can then detect them using the two-color approach. So we can really see these earlier species. If we then count the number of fibrils using the same approach, we can see that we also detect fibrils. So this staple approach allows us to detect fibrils and early oligomers. So we're detecting a wider array of molecules using this all different aggregates. What about specificity for different protein aggregates? Well, if we take our SAVE approach once again, and we look at no protein, we don't see many events. If we then look at amyloid beta, alpha-synuclein, or tau, we can't tell the difference between these. So they're all thioflavin T active. So there's not really, all we're doing is we're counting the number of them, but we can't say whether it's specifically amyloid beta, alpha-synuclein, or tau. All we get is thioflavin T active oligomers. If we then look at our staple approach for alpha-synuclein, we only see alpha-synuclein containing aggregates here. We don't see anything with the amyloid beta, the no protein, or the tau controls. We only see the alpha-synuclein aggregates. If we then do the same approach for tau, we don't detect any protein in the no protein sample. We don't detect aggregates in the amyloid beta sample, nor the alpha-synuclein sample. We only detect aggregates in the tau case. So what we've shown is that using this staple approach, not only can we detect the earlier oligomers, we can also detect specifically those oligomers containing the protein that we're interested in. And in this case, we've demonstrated it with alpha-synuclein and tau. But obviously, you could do this with any protein that you're interested in, be it TDP43 in ALS or other aggregating proteins in other diseases. So we're quite excited by this staple approach. We next wanted to then look in antemortem CSF samples, so cerebrospinal fluid samples. So what we did is we took antemortem CSF, and what we've done is we've taken 7 Parkinson's disease patients and 5 Alzheimer's disease patients with 13 healthy controls. And once again, we've used the UCB anti-alpha-synuclein antibody. And when we look at our PBS control, so without any CSF present, we're not detecting many events. When we then look at our healthy control, we detect a few aggregates, but not a huge number. You can see here we have some of them in the healthy control in grey here. And then when we look in the Alzheimer's disease, we're detecting some alpha-synuclein containing aggregates. But then when we finally look in the Parkinson's disease, we see that we get a statistically significant higher number of protein aggregates. So we're detecting more protein aggregates containing alpha-synuclein in the Parkinson's disease case compared to the healthy control or the Alzheimer's disease patient. So therefore, staple offers an exciting approach towards diagnosing these underlying proteinopathies. So we're specifically detecting protein aggregates made from a specific protein in biofluids from patients. However, if we were to take one of these purified oligomer samples that I showed previously, the ones from Stressmark, for example, and we were to put these on an AFM grid and image them using AFM, we would find that they're about 3 to 15 nanometers in size. However, when we image them using fluorescence microscopy, either using thioflavin T or our staple approach, everything looks to be about 250 nanometers in size. And the reason for this is because of the diffraction limit of light. So because light is wave-like in nature, we get constructive and destructive interference, and that leads to blurring. So what we next want to do is we want to be able to image these at a relevant spatial resolution. So we want to be able to get down to that tens of nanometer resolution rather than 250 nanometer resolution. And for this, we use super resolution microscopy. So in 2014, the Nobel Prize in Chemistry was awarded to Eric Betzig, Stefan Hell, and W. Murner for developing the super resolution techniques. So that blurriness that I showed you there is actually a probability distribution. If we imagine we've got GFP here and we image GFP, then our size of our point spread function, it's called, or our blur, is about 250 nanometers in size. Now this gives us some information. The most likely position that we would find our molecule is actually in the center of this blur here. So all we need to do is fit this to a mathematical function. If we fit this to a mathematical function to find the center point, we then know where our molecule is present. So this is like looking at Google Maps when you have your circle here. So quite often you would have your circle in the middle of where you're likely to be, and you'd have this blue circle around the outside. That blue circle is then your uncertainty in your GPS position. So we can find the center point. Now the problem arises if you've got two molecules close together, or in this case, two people close together. If two molecules are close together, rather than getting the center point of each one of them, if they're close together in this blur, we get the average position of the two. So it would be the average position of the two molecules or the two people standing together. So we wouldn't be able to resolve them. So what we do in super resolution microscopy is based on this idea by Eric Betzig. So he published this paper in 1994. And what he said is, okay, we've got all of these overlapping blurs here. If we could somehow separate them, either using the frequency or maybe time, and find the location of each one individually, we could then rebuild the image with all of those center points. And by doing that, we'd have a super resolution image. We'd be able to separate them at a greater resolution than the diffraction limit of light. So imagine this, we have two people standing next to each other at one point in time. But what we can do is we can switch one person off, or one molecule off and the other one on. We can find that location. So we find the first molecule or the first person. We then switch that one off and switch the second one on, and we find the second position, or we'd find the second person. And in that way, we've been able to resolve the two of them. So all we have to do with super resolution microscopy, or this particular type, is just make the molecules switch on and off in time. We make them blink. And as they blink, we then find the location of each one separately, and then build up a super resolution image of all of the individual locations. So this is what it looks like. This is just an example of, this is supposed to represent the nuclear pore complex. With all of the molecules on, conventional microscopy, we just see a blur. But what we do is we make them blink on and off separately, find the individual location of the molecules as they blink on and off separately, and that then allows us to resolve the position of each one of them, and we then get a super resolution image. So in conventional microscopy, we're limited to 200 to 250 nanometers, whereas in super resolution microscopy, we're limited to a resolution of about 20 nanometers. So one way that we can do super resolution, make the molecules blink on and off, is by using this paint technique, or this point accumulation for imaging in nanoscale topography. Now in paint technique, we have these molecules, in this case it's this dinyl red that's moving around in solution. Now quite often it's moving around so quickly that we can't resolve them on the camera. And what happens is when it binds to a protein, or to a specific region of the oligomer in this case, it's temporarily immobilized. So it's moving around really quickly, being smeared out, but when it hits the oligomer surface, it stays still for a short period of time. We can then get a blink of fluorescence as it stays still, and we can then get the location of that blink. So that allows us to map the surface of protein aggregates. So this is just showing you what the raw data would look like. We have blinking as the NAL red binds to the surface. We find the location of each individual binding event. That then allows us to build up our super resolution image. So we can see here it's a small fibril. So in the standard image, we have a big blur, but in our paint image or our super resolution image, we can see it's a small fibril. Now, once again, NIL red isn't specific. So it's like THT, it could bind to any different protein. It could actually bind to any hydrophobic surface. So if we wanted to image protein aggregates in a cell, for example, we wouldn't be able to do that because the cell would bind to NAL red as well as the thioflavin, as well as the protein aggregates binding to NAL red. So we want to increase the specificity. And one way of doing this is with DNA paint. So DNA paint works in exactly the same way. We have this reservoir of fluorescent molecules, but in this case, rather than it being a free dye, the dye is attached to a DNA sequence, the so-called imager strand. And as these are moving around in solution, they're diffusing so quickly that we don't observe them. But our protein of interest, or our molecule of interest, is then tagged with a complementary sequence to this imager strand, the so-called docking strand. And what we see is temporary binding events as we get hybridization, and we can then find the location of those. So this is just the fibril that we're imaging. You can see binding as each one of these DNA strands targets our antibody here. In this case, it's an antibody to alpha nucleum. And we get the binding of the imaging strands. We can find the individual location of these, and that then allows us to build up a super resolution image. So we can do super resolution imaging using DNA paint. Now what we've done is we've taken our staple approach, and this is work done by Rebecca Salib again and currently unpublished. We're doing exactly the same process as staple, but this time rather than having two colors on the antibodies, we have two different docking strands. So we have a docking strand with one sequence, a docking strand with a second one, and we then have two different color imager sequences. We can then look for coincidence between the two colors, this time with the super resolution image, and that then allows us to detect oligomers separately from our monomeric protein. So this is what it would look like with diffraction limited in our conventional imaging. You see there's some overlap between the magenta and the blue here. But when you turn on the super resolution imaging, you can see we have a higher resolution. We can see these small oligomers, and we can actually see the structure of them. So we have small oligomers, we have long fibrils here, and we also have then some larger oligomers as well. So now we can use our staple approach to actually image at the super resolution level. So overall, I've shown you that we can detect individual molecules, so we can see protein aggregates that are labeled. We can then use label-free detection, so it could be thioflavin T to detect the oligomers. We've increased the specificity by using the staple approach, which allows us to detect proteins specifically within aggregates. And we've then used a development on this using DNA paint, where we can do super resolution imaging of these protein aggregates. So I'd like to thank you for taking the time to watch this presentation. I'd also like to thank the people who are mainly involved in this. So that's Rebecca Salib, Gian Lee, and Craig Layton, and Judy O'Shaughnessy here. And the major collaborators on this work were Sonia Gandhi and Thilo Kunath. I'd also like to thank Stressmark Biosciences for providing the proteins that we use to image in this, and also UCB Biopharma for funding the project, and also for providing the antibodies that we used within this. I should also say that it does work with commercial antibodies as well. So we've shown that this works with UCB's antibodies, but also standard antibodies such as the Michael J. Fox antibodies. Okay, thank you.